Welcome to an overview of the endosymbiotic theory, the idea that complex eukaryotic cells evolved by incorporating free-living bacteria. In this slide, you see a simple diagram showing a host cell taking in a bacterial partner and gradually transforming into a cell with internal organelles, which sets the stage for how mitochondria and plastids first appeared in evolution. This slide traces the revival of the concept and the evidence that brought it back to the forefront. In 1967, the idea was reintroduced, and later molecular phylogeny and biochemistry showed mitochondrial and plastid genes clustering with bacterial lineages, which transformed endosymbiosis from a speculation into a central principle of modern cell biology. Here, we detail genomic features that point to a bacterial origin for mitochondria and chloroplasts, such as circular DNA, prokaryotic 70S ribosomes, unique tRNAs, and genomes lacking histones and introns. The comparison with bacterial cells and contrast to the eukaryotic nucleus strongly suggests descent from free-living bacteria rather than de novo creation inside the host. This illustration explains how phagocytosis and membrane architecture support the theory. A host cell engulfs a prokaryote, creating a double membrane around the symbiont. The inner membrane chemically matches bacterial membranes, while the outer membrane derives from the host. And together, these structural clues, backed by genetic data, explain how mitochondria and chloroplasts retain prokaryotic features. Phylogenetic analyses, shown here, pin down ancestral relationships. Mitochondrial genes cluster with alpha proteobacteria and plastid genes with cyanobacteria. Importantly, this clustering is independent of the host nuclear DNA, confirming that organelles share direct bacterial ancestry and are not simply rearranged host genome components. This slide walks through the 4 stages of symbiosis and integration, from initial phagocytosis and trapping to stabilization through metabolic exchange, then genome reduction and transfer of genes to the host nucleus, and finally, stable cellular integration, where the symbiont becomes an organelle, locked into a new partnership with its host. Secondary endosymbiosis explains how photosynthesis spread across diverse lineages. A predatory eukaryote engulfs a photosynthetic alga, keeping its plastid plus membranes, which leads to complex plastids surrounded by additional membranes. The images illustrate how these repeated events produce the diverse photosynthetic lineages we see today, like diatoms and dinoflagellates. Endosymbiosis massively accelerated evolution by freeing up energy through aerobic respiration and photosynthesis, which supported larger genomes, more elaborate cytoskeletons, and the emergence of multicellularity. This energy surplus permitted rapid diversification into many ecological niches, fueling the broad adaptive radiation of eukaryotic life. Understanding endosymbiotic origins has practical implications for medicine, agriculture, and synthetic biology. Recognizing organelle dysfunction helps explain metabolic and neurodegenerative diseases. Plastid engineering can boost crop yields, mitochondrial DNA informs targeted therapies, and chloroplasts can be repurposed to host new metabolic pathways in plants. Comparative genomics maps the roots of gene transfer and the limits of genome reduction, highlighting innovations like protein import machinery and functional adaptations. These insights guide synthetic endosymbiosis approaches, where engineered DNA and synthetic biology models aim to create novel organelles or integrated cellular systems for new energy and metabolic capabilities.